Before explaining what DNA actually looks like and the structure of it, we need to just understand the sort of naming system behind the carbon atoms on DNA. So there are carbons within this adenine, but they're not of importance to us. And there's no carbons within here. So we need to focus on the carbons within the deoxyribose sugar. So we have carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. And when naming the strands of DNA, we talk about the 3', which is here, and the 5' end of each nucleotide, which is actually effectively this hydroxyl group. Basically, these are the bonds that we can use to bind DNA nucleotides together or just to other things. And when we look at the structure of DNA, it will become more obvious what I mean by that. So let's actually go in and look. So it's actually numbered for us, which you see it very well. If we wanted to grow this DNA strand, so if I wanted to add another nucleotide onto this DNA strand, I could either add them here, because the hydroxyl groups are the reactive bit, so I could either add it here, or I could add it here. So this means that you actually have two ends to your molecule which are asymmetrical. You've got the 5' end, which is sort of the bit with the phosphate exposed, or you can have the 3' end, which is the end of the molecule where the 3' end, the 3' hydroxyl group is exposed from the actual deoxyribose sugar. If you look on the other strand, you'll realise the 3' end is facing upwards, so is aligned with the 5' end, and the 5' end is aligned with the 3' end. Basically, this is where the word antiparallel comes from. So this is why DNA is described as antiparallel. The backbone of, as we mentioned before, this sugar phosphate backbone, here, okay, this bit, is formed by the formation of phosphodiester bonds between two nucleotides. So if that's one nucleotide, that's another nucleotide, you have the formation of a phosphodiester bond here between the two. And it is a covalent bond. It's what gives the phosphate sugar backbone its sort of strength that is notorious about it. On the 1' carbon, you've got your nucleotide, you've got your bases, so your nitrogenous bases. And obviously you always have T opposite A and you have G opposite C. But you need to know more detail than that for A level, and you need to know about your purines and your pyrimidines. The easiest way to remember this is that a pyrimidine always pairs with a purine, so A always goes with T, and C always goes with G. So one of these is a purine, one of these is a pyrimidine in each group. I'm actually going to draw it the other way around. So G always goes with C, and Ag is the chemical symbol for silver, and a key property of silver is whether it is pure or not. So these are your purines, and your purines have two carbon and nitrogen rings within them. A and T form two hydrogen bonds between them, and G and C form 3. These hydrogen bonds are what keep the two strands together, and what is broken when you are trying to replicate DNA or to transcribe DNA to form mRNA. In terms of what you need to know, that is basically it in terms of for A level thus far.